I'm very excited to be here and tell you about BBC. It doesn't mean British Broadcast Company, but Brain, Body, and Consciousness. So consciousness is a big word, but it's at best a very fuzzy concept. And the study of consciousness remained in the realms of philosophy until more recent advances in non-invasive brain imaging and also experimental stimulation paradigms allowed us to study the neural correlates of consciousness. So how do we do that in the lab? With healthy humans, we can experimentally dissociate sensation from perception. What does that mean? It means we can look at stimuli where identical physical stimuli give rise to different perceptual outcomes. For example, ambiguous stimuli. I think everybody here is familiar with the Necker cube. Can face this way or that way. Probably you're also familiar with Rubin's vase stimuli. You can either see the vase or the two faces, both are present. Who sees the lady's face? Please raise your hand. Who sees the little dude playing the saxophone? Very good. Who sees a Canadian flag? Who sees two angry guys shouting at each other? Here's one, and there's the other. Another way of doing that is presenting stimuli at the sensory threshold, meaning you perceive the stimulus in 50% of cases and you miss it in 50% of cases. And one way of doing it is to use masking. Here's meta-contrast backward masking. Or another way of doing that is you can have stimuli. Here's a Gabor grading oriented to the left. I hope everybody sees this. Embedded in random dot noise. So you can either see the Gabor or just see the noise. And then we can just compare the neural response for different perceptual outcomes of the same stimulus. So when we measure neural correlates of consciousness, we have the stimulus here, we have different perceptual outcomes here, and there are two complementary ways of studying them. On the one hand, we can look at stimulus-evoked differences in neural activity. Right? We can just compare the response of the brain when we see the stimulus versus when we miss it. But given that this is always the same physical stimulus that sometimes is seen and sometimes not, the fate of the stimulus has to arise from the state of the brain. So we can look at pre-stimulus differences in the brain state that gives rise to different perceptual outcomes. These are just complementary ways of studying that, and it's not like that one is better than the other. Now, what has to happen in the brain for us to become aware of a stimulus? So first of all, there's a feed-forward sweep in visual areas down the ventral and up the dorsal stream, and this is necessary but not sufficient for conscious awareness. Next, there is recurrent processing, so activity goes back and forth within visual areas that is required for conscious awareness. And then there is widespread recurrent processing outside visual areas in frontal and parietal cortex. And this global availability of information throughout the brain is necessary for us to say, I saw it. But actually the role of frontal and parietal cortices for conscious awareness is nowadays disputed. So according to the global workspace model that says that information has to be spread throughout cortex, it has to reach frontal cortex, they really put emphasis on prefrontal cortex for awareness. Now, more recently, it has been proposed that a posterior hot zone, so activity in posterior parietal cortex, is crucial for awareness. So in situations where you have a stimulus that you sometimes might and sometimes might not see, only when you execute a response, you get activity in frontal cortex. When you do the same experimental paradigm but do not require an overt response, you see more parietal cortex. Another brain area that is really important for awareness is the insula. The insula is sort of a brain inside the brain, as a neurologist friend, I think, very pointedly put it one day. And it's also it's the so-called visceral cortex. So posterior insula receives input from the viscera, from the inside of the body, and it has a fundamental role for interoception. So if you try to listen to pick your favorite organ, your stomach, your guts, your heart, that's where the information from these organs is represented. And it has a posterior-anterior gradient from more interoceptive to exoreceptive processing. And the insula makes vast anatomical connections. So the posterior insula is connected to parietal and temporal cortex, whereas the anterior insula is anatomically connected to frontal cortex. The insula is together with the ACC also forms the main hub of the so-called salience network. So when you become aware of something, of the salience of a stimulus, these areas become involved. And you could think of the insula as like a multimodal integration hub that is involved in awareness. So when we want to study brain measures of the neural correlates of consciousness, and we want to distinguish between stimulus-evoked differences and pre-stimulus differences, we need a measure that has high temporal resolution that can actually distinguish in real time or with high temporal resolution what comes before the stimulus and what comes after the stimulus. And for that, we use EEG. EEG is the oldest window into the human mind. It's been around for 102 years. And this is one of Hans Berger's subjects with the electrodes stuck on the surface of the scalp. These are the readouts. So we've come a long way. We've come from a vector to a matrix. And you can really think of the EEG in terms of the temporal evolution of the scalp electrical field. So all momentarily active intracranial sources, they generate an electrical field that is spread out over the surface of the scalp, and this is what we measure, the temporal evolution of the scalp field. So basically you can think of the EEG as a matrix that has time in one dimension and space in the other dimension. So this is just 12 seconds of EEG from 64 channels, and classically what we do is we pick an electrode and we look at the electrode in time. We can also look in the other dimension. So this is just a different rendering of these 12 seconds of EEG in terms of the configuration of the scalp electrical field. And what we see is, I hope you see it, the configuration of the scalp electrical field remains stable for brief periods. Here's a polarity inversion, here's a different topography, a polarity inversion. So unlike the local measures that change from time point to time point, the topography of the scalp field remains stable for brief periods between 50 and 100 milliseconds. And these periods of quasi-stable scalp topography are called the EEG microstates. And usually at rest in healthy, so actually not only in healthy subjects, there are 4 dominant topography that are observed throughout the lifespan. It's an overall measure of momentary brain activity that is reference independent. Now, if we have the topography of the scalp electrical field, in principle, we can also reconstruct the sources that generate this field. Because I'm going to show later on, I'm going to show EEG sources, I lose a few words about it. So in principle, we have the topography of the scalp field, and that can in principle be generated by an infinite number of combination of sources. That's a so-called inverse problem. It's ill-posed, to go from the scalp potentials to the active neurons. But we can solve it. Now we need a forward solution, we need a biophysical model about how activity in certain parts of the brain is generated, how it spreads, and how it generates the scalp field. So with the forward solution, we can solve the inverse problem. Now when we look at ERP signatures for conscious awareness, ERPs are just event-related potentials. It's the EEG time-locked to the onset of events, of stimuli, of comparable conditions. Which yields a series of components. In this case, we look at it in the time domain. So here we compare the response of the brain when we see the stimulus in dashed versus when we don't see it in solid. And what we see is there are early differences that reflect sensory processing. And this P1 component that reflects to the first feed-forward sweep generated an extrastriate visual cortex, but it's debated. Some people find it, other people don't find it. Keep that in mind, we'll find a solution. Next, we find mid-latency, differences that reflect perceptual processes, the so-called VAN. You find a component, you give it a name, visual awareness, negativity, and then there are late differences, post-perceptual processes. It's called delayed potential or late positive component. It's a P3B-like potential, target detection. And that corresponds to the more widespread availability of information. So yes, we know that there are stimulus-evoked differences in neural activity when you see something versus when you don't see something. So what about pre-stimulus differences in neuronal activity that are related to awareness? So the EEG alpha band, it's a activity between roughly 8 and roughly 12 hertz. It's considered the so-called idling rhythm of the brain, and that could be associated with perceptual awareness. So basically, higher alpha power means more idling, means less cortical excitability, and pre-stimulus alpha power is higher when we miss than when we see the stimulus. Now, we looked at EEG microstate. It's a global measure of brain activity, the momentary state of the brain. And here we could also show a double dissociation when subjects consciously, or when subjects correctly identified a stimulus with and without awareness. And that was related to differences in the state of primary visual cortex. So yes, the pre-stimulus of the brain can predict awareness. We can plug that in here. So we have clear ERP components that are associated with different stages of processing leading to awareness. And we also have both local and global EEG measures that can help us predict if we see something or not. So when we shift gears a little bit, when we talk about consciousness, we talk about the brain. Right? That's a little bit of a no-brainer. But the brain is actually really small. It's 1.5 kilograms. It's maybe 2% of our body's weight. It's also a very greedy organ. It eats 20% of our body's energy. So with every block of chocolate, 20 grams go straight to your brain. Now we can rephrase this in terms of are 98% of our bodies irrelevant for consciousness? Or in other words, again, is the brain the only one or one of several organs that can contribute to consciousness? And we're not going to talk about the bladder, but the little prince had a hunch. The little prince said, it's only with the heart that one can see rightly. What is essential is invisible to the eye. So why the heart? This is the first time I saw my oldest son. And I was not so amazed by this little blip of human, but what really blew my mind at the time was it has a beating heart. It went. Now, the cardiac rhythm is like the first pacemaker of the developing organism. The proto-heart beats long before the neural tube closes. And that rhythmic cardiac the developing nervous system already has a rhythmic input from the proto-heart. Now, another important bodily rhythm, it starts at birth, is respiration, right? Hopefully when we're born, we take our first breath, and that kicks off a critical physiological transition. So the organism goes from anaerobic fetal to aerobic neonatal metabolism. So before we extract oxygen from the blood, and the moment we are born, we extract oxygen from the air, and everything changes. The entire metabolism changes. And breathing is also a rhythmic input into the system. And breathing is interesting because breathing is it's the only physiological process that both unfolds automatically, but it can come under volitional and conscious control. It's a very important rhythm if you want to access and control rhythms in your body. So why is cardiac activity important? it's because of baroreceptor signaling. So this is the EKG. It's actually my EKG. It's a series of characteristic peaks and troughs. And the cardiac cycle can be subdivided in two phases. The systole, it goes from the R peak to the end of the T wave, which refers to the contraction of the heart. And the diastole, from the end of the T wave to the following R peak, which is the relaxation of the heart muscles. Now, during the systolic phase, baroreceptors are active. Baroreceptors are stretch sensors that regulate blood pressure, and they are located in the aortic arch, the aorta, the big artery that goes around the heart, and the carotid sinus. The carotid is the artery that supplies blood to the brain. And they signal to the brain through the aortic ones through the vagus, and the carotid ones through the glossopharyngeal nerve. And they terminate in the brainstem, and then they project to the posterior insula. Why is this important? Because they relay an internal visceral signal to the brain. So roughly once a second, you get a signal from the baroreceptors to your brain, about every second throughout your life. And that modulates the processing of sensory stimuli. So simple visual stimuli, simple sensory stimuli are processed preferentially during the diastole when the baroreceptors are silent, and emotional stimuli are processed preferentially during the systole when the baroreceptors are active. So the cardiac cycle affects somatosensation. Here's a study from Al. They show that somatosensory threshold stimulus, like a faint electrical pulse to the index or the middle finger, they're detected better in the diastole towards the end of the cycle than in the systole. They're more likely missed in the systole. And they also elicit larger mid to late latency somatosensory evoked potentials in the diastole than in the systole. Now, when we look at when they looked at the pre-stimulus response to the heartbeat evoked potentials, it's bigger before misses than before hits. And also the SEP amplitudes are inversely related to the pre-stimulus HEPs. So in other words, the stronger the response of the brain to the pre-stimulus heartbeat, the more likely you are to miss it, and the smaller the response of the brain to the somatosensory stimulus. So what they put that in a predictive coding framework, and they proposed that the response of the brain to the more or less predictable heartbeat is suppressed or it's attenuated in order to improve the processing of the somatosensory stimulus. Keep in mind that the somatosensory stimulus and the heartbeat, they share parts of the neuronal pathway. The idea is, okay, when they compete along the pathway, we attenuate the response to the heartbeat in order to better perceive it. Now, respiration is, or the primary physiological function of respiration is gas exchange. We breathe in O2 and we breathe out CO2. But it's become a very popular rhythm in road electrophysiology. And it has been shown that neuronal network dynamics are coupled to the respiratory phase. So what we see here is the respiration and LFP activity in medial prefrontal cortex, and you can see it with your bare eye that they are coupled both in the time and in the frequency domain. Respiration also modulates broadband resting state activity. What we see here is MEG activity as a function of this inspiration and expiration. On the y-axis is frequency, and on the z-axis color-coded is basically the coupling mode. And we see that broadband activity is coupled differently to the respiratory cycle as a function of frequency. But MEG activity, resting state broadband activity, is coupled to respiration. And there are larger VEPs, visually evoked potentials, during inhalation than during exhalation. So yes, respiration also appears to be an important modulator of brain activity. But cardiac and respiratory activity are also coupled through respiratory sinus arrhythmia. Basically, the cardiac frequency is coupled to the respiratory phase such that your heart beats faster when you breathe in than when you breathe out. This is just geared towards physiological efficacy. And this is a pretty complex phenomenon. It's, on the one hand, it's driven by the balance between the sympathetic and the parasympathetic nervous system. So when you breathe in, you recruit more the sympathetic nervous system. When you breathe out the parasympathetic, it's like, and that's also why respiration meditation works so well. When you breathe out longer than when you breathe in, you stimulate the parasympathetic nervous system more than the sympathetic nervous system, and your heart rate goes on. It's not like spiritual at all, it's just hardwired physiology. And then also by baroreceptor fluctuation. So during inhalation, baroreceptor activity goes down, heart rate goes up. When there's more oxygen available, you use it better, and the opposite is during exhalation. So the question is now, is the effect of the respiratory phase via a direct mechanism, so the stimulation of olfactory bulb neurons, or is it indirect through baroreceptor-mediated mechanisms, or is it both? Another way of thinking how baroreceptors affect cortical processing is through gain control. So baroreceptors modulate cortical excitability via gain control. Basically, the amount of input signal that you need in order to cross a certain threshold. So when there is a distractor signal, in this case we have the visual signal and the visceral signal, when it does not have to be attenuated, you need less input. The cortex is excited at lower input intensities than when you have to attenuate the competing visceral signal. So baroreceptor activity is low during the diastole and during inhalation, so we have high gain and high cortical excitability. And the reverse is due for high baroreceptor activity in the systole and during expiration, so there we have low gain and low cortical excitability. So the question is now, how do bodily signals shape awareness? Is it a central attenuation of the brain to the predictable heartbeat, as has been proposed? And the question here is it a general phenomenon, or is it restricted to somatosensation when they share the neural pathway? In that case, it's late but not early SCPs that are modulated. But this idea makes no prediction about the respiratory phase. Now, if it's baroreceptor-mediated gain control, the picture looks very different. Because we should, if it's really cortical excitability, we should see a modulation of early ERP components, but only for low baroreceptor activity. They should be modulated when there is no competing signal coming in from the body. And if that is true, we should find modulation of mid-latency and late ERP components, irrespective of baroreceptor activity. That should only affect the early components. And it makes comparable predictions for the cardiac and the respiratory phase. So how do bodily signals shape awareness? Well, baroreceptor activity modulates cortical excitability and gain control. And the respiratory phase, respiratory activity can modulate awareness once via synchronization of brain activity through olfactory bulb stimulation or through baroreceptor activity. So what we want to know now is, how do bodily signals affect electrophysiological correlates of awareness? We're going to look at whether cardiac and respiratory phase affect stimulus-modulated brain activity, the ERPs. And we're also going to look at whether the state of the body affects this brain state-modulated stimulus processing. We tie this all together into a terribly simple experiment. Subject saw a Gabor presented for 16 milliseconds. That's very, very brief. Actually, for skiers, that's a very long time to lose. If you think of it, like 60 milliseconds, it's really nothing. So they had to make an orientation discrimination of whether face is left or right, and then they had to give a subjective awareness judgment. And we wanted to compare the same physical stimulus when it was correctly identified. Physical stimulus is the same, the response is the same, and the only difference that we look at is subjective awareness. So we aimed for at least 75% correct and 50-50 correct, aware and unaware. And then we just looked at the effect of the cardiac and the respiratory phase on these awareness-related ERPs. So we had 120 channels of EEG. We measured the EKG and respiration. We had 30 subjects included in the final analysis, and we classified the trials according to the cardiac and the respiratory phase in which they fell. Now, the diastole is much longer than the systole. And so we only considered trials that fell into the window in the end of the diastole that corresponded to the duration of the systole in that cardiac cycle. Because you don't want 1.5 more trials in one condition than in the other. And pretty much all of the heart rate variability is due to the variability in the diastolic phase. So when we look at the behavioral results, actually we had 85.5 correct trials. They were very good. And we had 50.4 and 49.6 aware, unaware. This is very difficult to do. We can talk about that later if you want. And the subjects are faster when they see than when they miss the stimulus. But in the reaction times, there was no effect of the cardiac or the respiratory phase on reaction times. This is due to the fact that they were at ceiling. And we aimed at having high accuracy. When we plug in the errors, we do find a trend towards an effect. So they are faster, tend to be faster in the diastole than in the systole. But what we did not want is they say, I saw the stimulus, but they give the wrong answer. We had some highly overconfident subjects. They always say they saw it, but they were just a chance. They didn't see anything, but they said they saw something. So, voila. So when we look at the main effect of awareness, we find the P1 modulated, we find the VAN modulated, we find the LP modulated. Nothing new, but it's always good to show. Now let's look at it in more detail. Now it gets interesting. So we do find the P1, as you can see just with your BI, the effect is rather small. So the earliest marker of awareness, we do find the P1, but only in the diastole, but not in the systole. This is the P1 effect. You see it here, you see it here. It's attenuated in the diastole when subjects miss the stimulus, and the same thing for inhalation. We find the P1 during inhalation, but we do not find it during exhalation. And we find the VAN for active baroreceptors. So here we plot this is the VAN. Here I plot the effect size, and it's the earliest component that is modulated during the systole, and the effect size is also bigger than during the diastole. Same thing holds for the respiratory phase. We do find the VAN as the earliest marker during the systole. And that supports this notion of baroreceptor-mediated gain control, because we do find a modulation of the sensory component when the baroreceptors are silent, when there is no competing signal from the body. And that solves also this controversy of whether the P1 is the earliest marker of awareness or not. Well, yeah, it depends if there is a signal from the body that comes with it or not. When we look at the sources of the P1, we do find a higher current density in excess striatovisual cortex in the diastole than in the systole when the subject sees the stimulus. And this, again, it speaks for this idea of higher gain or higher cortical excitability during the diastole. Now, when we look at the sources of the VAN, it gets interesting again. So here, it's a pretty long component. It has a rising and a falling phase. And then when we consider the entire time window, we find a posterior insula and temporoparietal junction. Then we subdivided it into the rising and the falling phase. And we find that in the rising phase, we only find differences during periods of high baroreceptor activity during the systole and exhalation. So here we find the posterior insula and also the TPJ. And our claim is the the signal from the heart, or the signal from the baroreceptors, is processed in the posterior insula, and then it is separated and segregated from the visual stimulus in the TPJ, the temporal-parietal junction. It's also a multimodal integration area. And this is where the separation happens, and only when the separation is successful can you say I saw it. Now, the anterior insula and prefrontal cortex, they are activated in the falling phase of the VAN, and that's sort of the integration of the visceral and the visual signals. So what does that tell us about early neurocorrelates of consciousness? So when the baroreceptor activity is low, the P1 is the earliest marker. When there's no signal from the body, early sensory components are modulated. And here is this baroreceptor-mediated gain control is the most parsimonious explanation. When the baroreceptor activity is high, the VAN is the earliest marker. And the interoceptive signals are processed in the posterior insula and then separated from the visual input in the TPJ, which is a necessary prerequisite for awareness. And then later on, the anterior insula redirects saliency towards the stimulus. Now, when we look at the sources underlying the LP, the late positive complex, the P3B, target detection type component, when we look across the board, we do find posterior and anterior insular, parietal, and frontal cortex. Oh, I have plugged it in.
Oh, is the plug on? Turned off.
Ha. No, should be fine. Holy moly. I got a very fancy schmancy adapter plug so I can at least use it. Very good. Whew. Thank you. Happened once in an exam. Students had to do the exam on a computer, and I didn't realize you had to plug in the projector for the socket to work. Okay, let's go back. So we find anterior, posterior, insular, frontal, and parietal cortex. Now, again, when we subdivide the effects again as a function of the cardiac and respiratory phase, we find that the posterior insula is reactivated when there is a signal from the body, and then activity spreads to the dorsal projection targets and parietal cortex. Now, when there is no signal from the body, the anterior insula is reactivated and activity spreads to prefrontal cortex. So both the posterior and the anterior insula get recurrently reactivated, and then activity spreads to the anatomical projection targets in parietal and frontal cortex. So the frontal cortex is only activated when baroreceptor activity is low. Well, we can solve another controversy, namely that of the role of frontal and parietal cortex for awareness. In our case, the subjects always had to make a response. But it depends. When there's a signal from the body, it goes through the posterior insula into parietal cortex. When there is no signal from the body, it goes through the anterior insula into frontal cortex. So I think I could show that baroreceptor fluctuations across the cardiac and the respiratory phase, they shape the earliest marker of awareness and the trajectory of brain activity that leads to awareness. Neither frontal nor parietal cortex per se are necessary for awareness, but they contribute independently and they adapt dynamically along with signals from the body. There is evidence for gain control to modulate awareness. The question is now, are baroreceptor fluctuations sufficient for gain control or not? And in nasal breathing, we have both, right? We have olfactory bulb stimulation and we have baroreceptor activity. Next, we wanted to know whether the effects of the respiratory phase are direct or indirect. Direct is synchronization through olfactory bulb stimulation. Indirect is baroreceptor modulated gain control. It could also be a combination of both. So during nasal breathing, we have both direct and indirect effects, and during oral breathing, we only have indirect effects. Baroreceptors are active. There is no olfactory bulb stimulation when we breathe through our mouth. We achieved that by very lightly putting a piece of surgical tape over the nostrils or over the mouth. We did not tape their mouths shut. So what we see is oral breathing differentially modulates awareness-related ARPs. There is no P1 modulated anymore. When we breathe through the mouth, same subjects, same task, different day. The earliest marker is the P2, and the P3 is modulated in addition to the VAN and the P3B. Now let's look at that in a little bit more detail. So there are no sensory components whatsoever modulated as a function of the respiratory phase. When we look at it for exhalation and inhalation, there is nothing, no sensory components. Baroreceptor fluctuations have no effect now that we breathe. The P3 is modulated only in the diastole, so it's a little bit later, and it's the baroreceptor activity is more tightly coupled in time to the cardiac than the respiratory phase. And the VAN and the P3B here, they are modulated irrespective of the cardiac and the respiratory phase, very similar to what we saw during nasal respiration. And the P3A is modulated only for low baroreceptor activity. It is generated in prefrontal cortex, and there is decreased oxygen availability in PFC during oral breathing. So it might be really related to the way oxygen is supplied to the brain, that this component is modulated in addition when we breathe differently. So the mode of breathing affects awareness-related activity. Breathing in through the mouth, the nose, and to the mouth, it's not the same thing. It does get air into your lungs, but it affects the brain very differently. So for nasal respiration, the P1 is the earliest marker, and for oral respiration, it's the P2 in the diastole and the VAN. So it looks like that this baroreceptor modulated gain control actually requires olfactory bulb mediated synchronization. And that supports the notion of at least nasal respiration as like an important oscillatory scaffold that can functionally couple brain activity across different areas. So yes, baroreceptor fluctuations per se are not sufficient for gain control. The sensory ERPs are not modulated when we breathe through the mouth. And it requires neural synchronization through OB stimulation. That's it before. So now we want to look at, yes, we've shown that the cardiac and the respiratory phase and the mode of respiration affect stimulus-modulated brain activity. And now we want to see whether the state of the body also affects the brain state-modulated stimulus processing. So basically, we just looked at the pre-stimulus microstates in the nasal experiment. We find 5 different microstates, and they differ for the respiratory phase. So microstate A is more present during inhalation than exhalation. It is associated with increased arousal. And microstate D, this one, is more present when subjects miss than when they perceive the stimulus. It's more present in the unaware than in the aware condition. So microstate D, we did a simultaneous EEG fMRI experiment where we could associate microstate D with the attention reorientation network. And it's negatively associated, so more of microstate D means less of attention. But the microstates do not interact with the cardiac or the respiratory phase. It's not like that one brain state preferentially occurs during the one or the other cardiac or respiratory phase. So they're independent. So basically the momentary state of the brain and the momentary state of the body, they exert an independent influence on awareness. So if I have a little bit more time, I can also talk about, we started a bit late, it's 5:15.
5 minutes.
5 minutes? Yeah, I can do it in 5 minutes. We also want to look at, to make the picture around, if we can predict awareness from the pre-stimulus response of the brain to the heartbeat. So for VEPs, there are higher pre-stimulus HEPs before hits than misses, and for SEPs, we just saw it's the opposite. So we want to see if we can also predict awareness by looking at the pre-stimulus AGP. And also we want to see whether inter-individual differences, which are also often considered as noise, play a role in that. So there's one problem, big caveat. So the VEPs and the AGPs, they temporally overlap. There's always an AGP. And every now and then there is a VEP. But the VEPs are much bigger than the AGPs. The thing is the following. The response of the brain to the heartbeat is independent of the response of the brain to the subsequent stimulus. In the brain, they go separate ways, but the signals mix. So far, what people have done to overcome this, they only looked at stimuli falling into the late diastole. But we've shown that ERPs differ for stimuli in the systole and the diastole. But if really the response of the brain to the heartbeat predicts awareness, it shouldn't matter whether the stimulus occurs in the systole or in the diastole, right? So one way of doing that, we need to unmix the signals. We need to apply deconvolution. Basically, we do a regression and we separately model, we do a time expansion of the matrix, we separately model the heartbeat evoked response and the stimulus predictor. Very simple. So it works. This is the regression-based AGP when applying deconvolution in the solid and not in the dashed line. And they're very, 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 very different AGPs preceding stimuli falling in the systole and stimuli falling into the diastole. And these differences, they occur across the board, across time and space. Now, this just illustrates how the signals mix in the systole and the diastole. And when we apply deconvolution, these differences, they miraculously disappear. So we really need to apply deconvolution if we want to look at pre-stimulus AGPs. Very important. Also, what many people show is pre-stimulus AGPs looks very, very conspicuously like sensory evoked potentials. For auditory evoked potentials, it's on the central electrodes. For visuals, more on the back, on the side. Voila. So when we apply deconvolution, we find differences over left frontal and occipital electrodes, but not over right temporal electrodes that other people have shown before. When we apply, when we use the regression, but we do not apply the deconvolution, we also find right temporal clusters that others have identified before without using deconvolution. And then when we do classical averaging of the pre-stimulus AGP, we find similar topographies than we find for regression without deconvolution. So these right parietal, temporal parietal differences that others have reported also for visual stimuli, that's residual stimulus evoked activity, but not heartbeat evoked activity. So in the last step, we wanted to see whether your ability to listen to your heart affects this. If you try to listen to your heart without touching a surface, it's really, really difficult. Some people are quite good at it. Some people are really bad at it. So basically, this is the performance in a heartbeat counting tasks. And we did a median split. We used the high heartbeat perceivers and the low heartbeat perceivers. In its right, and we did an ROI analysis in the electrodes that we identified before. So when we apply deconvolution, there is no difference in this right temporal cluster. When we look at the occipital effects, we do find a main effect, but it is entirely driven by the high heartbeat perceivers. Only if you're good at listening to your heart can we predict awareness from the response of the brain to the pre-stimulus heartbeat. When we do not apply deconvolution, we find these right temporal clusters, and that varies irrespective of interoceptive ability. But this occipital cluster, again, it varies. We find it only, it's solely driven by the high heartbeat perceivers. So yes, we can show that inter-individual differences in interoceptive ability determine if we can predict awareness from the pre-stimulus heartbeat. To wrap up, yes, the pre-stimulus HPs differ for subsequently seen versus unseen stimuli. We really need to apply signal deconvolution to disentangle the response of the brain to the heart and to a subsequent visual stimulus. And this is driven by inter-individual differences. It's only the ones who are good at listening to their heart that we can predict if you can see it from the response of the brain to the heart. So yes, we could say the state of the body does affect brain state modulated stimulus processing. To take home, bodily signals modulate awareness-related brain activity. They affect stimulus-modulated brain activity, determine the earliest electrophysiological marker, the trajectory of brain activity. They affect brain state modulated brain activity and exert an independent influence from the state of the brain. So the state of the body and the state of the brain, they exert an effect, but it's independent. It's not like they are coupled. And inter-individual differences in interoceptive ability determine if we can predict awareness or not. So bodily signals, they are commonly dismissed and eliminated as noise, similar to the way trial-by-trial variations are eliminated by averaging or regression. But cardiac and respiratory signals, they are constant drivers of the organism. And here we can show that they actually strongly modulate brain activity. We can, I don't know, I also considered myself like really encephalocentric brain snob. And we think of the brain as a closed system. And we can study it as a closed system, but as soon as we look a little bit outside the box, we can show that bodily signals are very important for brain activity. It also goes nicely with Nikki's idea of like the embodiment determines if you fall for certain magic tricks and not others. And we really propose they should be recorded, considered and recorded as functional signals that modulate how the brain becomes aware. And that helps us to understand better the intricate coupling between the brain and the body. And a big thank you to my co-authors, and a particularly big thank you to Viviana Leupin, who actually did all of the brain-body studies, and the people in my lab over the course of these projects. And thank you for your attention.